This is our Cell Energetics project by Ivy, Kate, Mateen, and Hudson. Photosynthesis and cellular respiration are key processes to all life on Earth. In this video, we will go over how photosynthesis and cellular respiration work. Photosynthesis is a process in which plants create energy by turning sunlight, water, and carbon dioxide into glucose and oxygen. Photosynthesis involves two primary sets of reactions: light-dependent reactions and light-independent reactions, also known as the Calvin cycle. Photosynthesis is a necessary process for primary producers and ecosystems as a whole.
The equation of photosynthesis states that carbon dioxide and water in the presence of light becomes sugars and a byproduct oxygen.
The light-dependent reactions occur in the first part of photosynthesis. They use sunlight to create ATP, NADPH, and oxygen. The thylakoid contains a cluster of chlorophyll and proteins known as photosystems. Photosystem 2 is the first step in the photosynthesis process. In photosystem 2, light energy enters the thylakoid and splits H2O into hydrogen gas, hydrogen ions, and high-energy electrons. Chlorophyll molecules in photosystem 2 absorb light. This absorption of energy charges the electrons and gives them energy. Electron carriers then shuttle these high-energy electrons in photosystem 1. In photosystem 1, after photosystem 2, these electrons get re-energized with light. The difference in both hydrogen ions filtrations through the membrane is what provides energy to make ATP. The light-dependent reactions produce oxygen, which isn't used in further processes, and convert ADP and NADP+ into the energy carriers ATP and NADPH, which are then used in the light-independent reactions to make glucose.
In the light-independent reactions, carbon dioxide enters the Calvin cycle, combines with a 5-carbon molecule, and then splits into 2 3-carbon compounds. Energy from ATP and high-energy electrons from NADPH are used to produce high-energy sugars from carbon dioxide.
Cellular respiration is a process in which glucose molecules are broken down into around 36 molecules of ATP energy. Cellular respiration is the main process that autotrophs and heterotrophs undergo to obtain energy necessary for their cellular processes. Cellular respiration and photosynthesis are very similar in that they are opposite, with opposite reactants and products. While photosynthesis uses sunlight, water, and carbon dioxide to create glucose, cellular respiration breaks down glucose molecules into energy for organisms to use.
The equation for cellular respiration states that glucose and oxygen become carbon dioxide, water, and ATP energy.
Cellular respiration starts with a molecule of glucose being broken down into 2 molecules of pyruvic acid and 2 net ATPs and NADH molecules in a process known as glycolysis. Glycolysis takes place in the cytoplasm of the inner liquid of the cell. Glycolysis does not provide much ATP, but it converts the glucose molecule into more usable pyruvate. Additionally, the process of glycolysis only requires 2 molecules of ATP to start.
The 2 pyruvate molecules are then transferred to the mitochondrial matrix where the Krebs cycle occurs. In the Krebs cycle, the pyruvate is converted into acetyl-CoA, where it undergoes multiple chemical reactions giving off electrons that are taken by NAD+ and FAD+ to the electron transport chain. After the acetyl-CoA has been stripped of some of its electrons in 2 carbon molecules, it becomes axolotl-silatate. The axolotl-silatate is used again to react with the next acetyl-CoA that comes from the cytoplasm. This allows the Krebs cycle to continue indefinitely, which is why it is called a cycle. The Krebs cycle also produces 2 ATP and carbon dioxide as a byproduct.
Continuing, the electron transport chain takes the newly formed NADH and FADH2 from glycolysis and the Krebs cycle to the inner mitochondrial membrane. There, the electrons and hydrogen ions already in the mitochondrial space form a proton gradient. In the proton gradient, the hydrogen ions try to balance themselves out above and below the inner mitochondrial membrane and try to move towards a location so that the distribution of hydrogen ions is balanced. When hydrogen ions move across the membrane, it spins the ATP synthase. The ATP synthase uses this kinetic energy to synthesize ATP by binding ADP to a phosphate group. The electron transport chain produces around 30 to 34 ATP and water from oxygen and high-energy electrons. The oxygen is necessary to be the final acceptor of the energy-depleted electrons.
Fermentation is another way organisms create their energy. Very similar to glycolysis, fermentation breaks down glucose into ATP. However, fermentation includes another molecule so that NADH produced can be used and cycled back so that the process can continue. Like glycolysis, the process is incredibly inefficient compared to the full process of cellular respiration. However, it is useful as an emergency energy producer when organisms requires lots of energy or do not have access to oxygen. There are 2 main types of fermentation that organisms undergo: alcohol fermentation and lactic acid fermentation.